Hi guys and welcome. So in this video we are going to take a look at the 2020 Intermediate Biology Final Exam paper. On the right side of the screen you can see instructions that will be given to you on the first page of your exam worksheet before you actually start to do the exercises. In the presentation I indicated with abbreviations where the actual materials can be found in the textbooks that are connected to specific exercises. So the first book is referring to biodiversity and plant life, the second book is the cell biology, histology and human biology, and the third book refers to genetics, evolution and ecology. So let's look at the first exercise, animal identification key. Similarly to how plants are identified, work out step by step which animals are marked by A, B, etc. Write the name of the species next to the letters on the dotted line at the end of the task. You can see the animals listed and then the dichotomous key itself, which is really easy to use. So basically what you need to keep in mind is that you always start at the top and you are moving downwards. And when you read the first statement, that will lead you to the second statement that must be read. So then you read the second statement and that line will also lead you to the third statement that should be read. Then the third statement is going to give you a letter in this case, and basically in this exercise you have to figure out which animal will fit into this place according to the sentences you read. So let's look at the sentences. The gizzard is part of the animal's digestive tract. The body fluid in its blood vessels transport oxygen, and the oxygen-carrying hemoglobin is found dissolved free in the blood plasma. So the first statement is very selective because we know that out of these organisms, only two animals will have gizzard, and those two animals are the domestic hen and the earthworm. And basically, we can decide, okay, after the first statement, which one is going to be B. So let's read the second. The body fluid in its blood vessels transport oxygen. That's true for both, so it didn't really help. The oxygen-carrying hemoglobin is found dissolved free in the blood plasma. Now, this statement will help us to basically select which organism will be written to this dotted line, because we know that in case of earthworm, they basically lack red blood cells. They have a unique type of hemoglobin that helps them to transport basically oxygen, so earthworm. Okay, so let's look at the second animal. The animal has no gizzard, and it leads us to the first block. First statement, it has internal skeleton. All right, so let's see what is going to be D. Basically, two animals have internal skeleton out of these opportunities, basically the domestic hen and the carp. And we know that the hen has a gizzard, which helps to grind the food. So basically, carp is going to be the animal that must be written to the dotted line. Moving on with the third animal, 2B, the body fluid in its blood vessels does not transport oxygen. That's an easy one. It is going to be the forest caterpillar, hunter. Okay, 3B, the oxygen-carrying hemoglobin is found in the red blood cells. Domestic hen will come next to letter C. Moving on to the fifth animal, 4B, it has no internal skeleton, and it leads us to the fifth block. Partly or completely extracellular digestion takes place in the body of the animal. Then you need to jump to the sixth section, and its digestive tract consists of a foregut and the midgut. Now this statement is really helpful if you remember what you have learned about flatworms, and out of these animals basically the freshwater planarian can be written here. A highly branched midgut system you can observe in their bodies. Okay, the next animal is going to be E. So only intracellular digestion is carried out out of the organisms that are being provided, the freshwater sponge is the answer. Next to letter E. And the last statement, its digestive tract consists of a foregut, midgut, and the hindgut. And basically that is going to be the edible snail. Okay. Exercise 2 in the depth of lakes. The two graphs show the temperature and dissolved oxygen content of two lakes as a function of depth. 100% relative oxygen content is the oxygen content of the water, which is saturated with oxygen at the given temperature without any living organisms. The graph on the left shows a lake containing large amounts of dissolved nutrients, eutrophic, while the graph on the right shows a lake which is poor in nutrients, oligotrophic. First question. If all other conditions are kept the same, which lake is penetrated deeper by the light? Give your reason for your answer. So as a matter of fact, in case of a lake where the amount of nutrients are very small, basically it's impossible to support an abundant plant life, and planktons are not capable of preventing sunlight from penetrating deeper layers, as you can read it in the key as well. And obviously less floating decaying organic matters can be found in this kind of lakes, and that is going to be oligotrophic. Okay, and the explanation. Less plants can be supported by nutrients, this kind of lakes, and for example, planktons don't prevent sunlight. To reach deeper layers. Okay. What is the main source of oxygen in the lakes in addition to the oxygen gas dissolving into the water through the surface? Support your answer with an equation. Okay, easy one. Photosynthesis is the process you need to name, and the simplified equation of photosynthesis should be written in here. So 6 carbon dioxide plus 6 water in the presence of sunlight. Oops, sorry. Sugar plus 6 oxygen will be produced. Okay. What are the two main reasons for the decreasing levels of oxygen below the depth of 50 meters in eutrophic lakes? So the question is asking about this part of the first graph. And you have to keep in mind two things. The first one is the rate of photosynthesis in the nutrient-rich lake is lower at deeper levels because of the shading effects of plants. And the second one is connected to the consumer's cellular respiration. As we know, it is a process that requires oxygen. And obviously, this is also a contributing factor in this pattern that we can see that the oxygen levels are decreasing. So basically, we should write down these two things. Shading effect of surface plants and the cellular respiration of the consumers. Okay. For the autotrophic organisms in the lakes, the nitrogen and phosphorus containing compounds represents the main limiting factor. Name the compound in which the nitrogen is primarily taken up by these organisms. So here you can mention the nitrate ions or the ammonium ions. For example, if you want, you can use the chemical formula or you can write them down with words. But these are good answers. Where, in which type of lake, and in what range of water depth can the following organisms live in the summer period? Cod-loving species in between 6 and 10 degrees Celsius with high oxygen requirement, minimum 80%. So if we look at the first graph, we can see that these conditions are not met. But in case of the second graph, basically, the oxygen content is always higher than 80%. So this requirement is fulfilled. Let's have a closer look at the temperatures then. 6 is approximately here and 10 degrees Celsius is somewhere here. And we want to figure out what these points will give us in terms of water depth. It would be much better to use a ruler, but I will try my best to indicate it as accurately as possible. It is approximately 25 and 45 for me. In the key 2560 is given, but I just write 2545. So oligotrophic 2545 unit is important, meters. Warm-loving species minimum 18 degrees Celsius with high oxygen requirement minimum 80%. Okay, I use a different color. So minimum 18 degrees Celsius. So the temperature should be observed somewhere here and then somewhere here. And I know that the oxygen requirement is always higher than 80% in case of the second graph. So I just stick to this one and I try to figure out what water depth it is indicating. It's approximately 15 meters. But I have to tell you that the eutrophic lake is also a good answer in here. So oligotrophic, and the water depth is 0.15 meters. In the key it's 10.15. Species active in medium temperature ranges above 8 degrees Celsius and in waters with low levels of dissolved oxygen, maximum 60%. So immediately we can exclude the second graph, as we know that in case of the oligotrophic lake, the oxygen content is always higher than 80%. So let's observe the first one. More specifically, this line here is important, 60%, and the 8 degrees Celsius. Okay. And what does it mean? Basically, what is the answer here? The type of lake will be eutrophic. And the water depth, if you observe the graph, it is 15-25 meters. Okay. Continuing with the third exercise, vertebrates. Compare the three groups of vertebrate animals, write the appropriate letters in the box next to each statement. So we need to choose in between reptiles, birds, mammals, all three of them or none of them. Oxygen is taken up through the air tubules, parabronchi. That's an easy one. We know birds are doing double ventilation with the help of their air sacs. They have two-chambered heart. That would be fish, but it is not provided, so none of them is going to be the good answer here. Third statement, their body temperature depends on the temperature of their surroundings. So basically, out of the three vertebrate groups, only reptiles are cold-blooded animals. So A is going to be the good answer. They have keratinized skin. All of them have keratinized skin. In the larval stage, they breathe with their gills. That's not true. Yes, that's not true for these animals because amphibians have this magical characteristical feature, so E is going to be the good answer. They have internal fertilization. That's true for all of them. They have eggs with leathery shell, reptiles. And in most cases, the fetus stay in contact with the mother through the placenta. That's an easy one. Placental mammals have these characteristics. Looking at the fourth exercise in constant flow, a segment of the route the respiratory gases take in the human body is shown in the diagram. The flow of gases is indicated by the thick arrows. After studying the diagram carefully, write the appropriate letters in the boxes provided next to the statements, then answer the questions. First statement, the extracellular matrix of the blood. That's an easy question. Basically, C is indicating the extracellular matrix. It is formed in the red bone marrow. We know that red blood cells or erythrocytes are formed in the red bone marrow, so B. The gas moving towards the upper airways, A is showing us the gas which is moving upwards. It is made up of squamous epithelial cells, the wall of the alveoli itself, which is going to be D. It must be really thin in order to allow gases to penetrate through it and to allow efficient gas exchange. It is there that the lower airways led into. That's going to be D as well. And it is a gas released from cellular respiration, carbon dioxide. That must be A, as we can see that it is moving from the capillaries towards the alveoli or alveolus. Okay, from which chamber of the heart is the blood pumped directly into the pulmonary circulation? Write the letter of the correct answer into the box. In order to answer this question, it's important for you to remember what you have learned about heart and the circulatory system itself. So let's quickly recall. We have a little space in here. So our heart is made up of 4 distinct chambers: left atrium, left ventricle, right atrium, and right ventricle. The pulmonary circuit is responsible for connecting the lungs with the heart, and the systemic circuit connects the heart with the rest of the body. So from the body, the oxygenated blood will arrive to the right atrium throughout the inferior and superior vena cava. From the right atrium, it is pumped to the right ventricle, from where, throughout the pulmonary artery, the deoxygenated blood will travel towards the lungs, where gas exchange will take place. Oxygenated blood then will come back to the left atrium throughout the pulmonary vein, and this oxygenated blood will be pumped to the left ventricle, from where, basically, throughout the aorta, this blood goes towards the body cells. So knowing these, and going back to the 7th question, which is asking basically this pathway, we can simply say that the right ventricle is going to be the good answer here. Okay, let's look at question 8. To which chamber of the heart the blood flows directly after leaving the lungs? So question 8 is asking this pathway, and basically you need to name the chamber where the vessel will arrive, and that is going to be the left atrium, so C. What is the name of the process by which the respiratory gases pass through the wall of capillaries and alveoli? That's simple diffusion, so a passive movement of gases according to the concentration gradient. What causes the blood to change its color as it flows through the capillaries over the surface of the alveoli? So when oxygen binds to hemoglobin, basically the blood becomes oxygenated, and that will cause the color change. Question 11. The figure on the left shows the healthy alveoli, while the figure on the right shows the alveoli which are damaged due to smoking. Discuss the visible difference between the two and the consequences of it. Okay, so difference is quite easy to identify, that the surface area has been dramatically reduced in case of the damaged alveoli walls, and the consequence is also obvious. So the breathing surface is much smaller, therefore the gas exchange won't be as successful. So that's what we need to write down. And the efficiency of gas exchange is also reduced. Exercise 5, Magical Biology. After reading the excerpt below, which comes from the fantasy literature, answer the questions based on the information of the text and your own knowledge. Millie, the witch apprentice, is getting ready for her exam in Fantastic Beast Studies. Having read the description of 2 such creatures, she recalls what she learned at school in her biology classes. Basilisk, also known as the king of serpents, Harpo the Fowl was the first dark wizard who had the ability to speak the language of serpents, to breed a basilisk. After long experimentation, he accomplished this task by hatching a chicken egg beneath a toad, which resulted in a creature which reached a gigantic size and was more deadly than any other magical beast. The body of basilisk is glowing green and can be as long as 15 meters. The male specimens can be readily recognized by their scarlet red feathers growing on their heads. The basilisk can kill with its deadly and venomous fangs, but its most dangerous weapon is its murderous stare, and all who are fixed with the yellow beam of its eyes shall suffer instant death. Billywig. Billywigs are common insects native to Australia. Due to their speedy flight, they are rarely noticed. Even wizards and witches only notice them once they have been stung. The billywig's wings are attached to the top of its head, rotate extremely fast, spinning the billywings along with its other body parts so that it can fly. The billywig also has a long, thin stinger at the bottom of its body. Anyone stung by a billywig would suffer dizziness followed by hallucination and levitation. This is also the reason why young Australian wizards and witches have had the custom of collecting and provoking the creatures into stinging them so that they can enjoy the above-mentioned side effects. Which statements are true for the reproduction of a real toad, which belongs to the amphibians? Write the letters of the correct answers into the boxes. The toad has external fertilization, and that's something which is true. The toad needs no water for its reproduction because of its terrestrial life form. That is definitely incorrect. Toads do not go through metamorphosis, they go through metamorphosis, so this statement cannot be accepted. The larval form of the toad is the tadpole. That is definitely true. Toads are not typically separate-sexed animals. This cannot be accepted, so A and D are going to be the good answers. Which statements are true for the respiration of a real toad? In some of its developmental stages, the toad breathes with gills. This is true. The skin of the toad has an important role to play in gas exchange. Yes, they have skin breathing. The epithelium of the toad is heavily keratinized to help breathing. That is definitely incorrect. The intercostal muscle and the diaphragm are involved in breathing movements. Obviously not. They are actually swallowing the air. That's not the scientific way of expressing it, but that's the easiest way to understand. The respiratory surface in the toad's lungs is increased by alveoli. No, that is for the mammals. So basically the good answers will be A and B. Give you a reason why a toad would not be able to hatch a chicken egg. We know that the toad is a cold-blooded animal and the chicken egg needs warm environment in order to hatch. So let's use the scientific terminology. Toads are poikilothermic, which means that they are not capable of maintaining a stable internal body temperature. And that's the reason why it's impossible for them to hatch a chicken egg, because they can't keep the eggs warm. Okay, so we write down here that toads are poikilothermic. They can't keep the eggs warm. According to the author of the text, the basilisk has feathers similar to those of birds. Although such creature only exists in fantasy, the reptiles and birds are related to one another. Name one such feature which is shared by both reptiles and birds, but not by amphibians. Okay, so we need to compare reptiles and birds and amphibians. Reptiles and birds have internal fertilization, while amphibians have external. So we write here internal fertilization. Internal fertilization. And also you can think about the amphibian skin breathing. And in case of the reptiles and birds, they have keratinized epithelium. As the text suggests, the venom of billywig is used by some as a psychedelic. The altered state of mind appears to be a desirable condition in the texts. However, the real drugs, which do not have this effect, present a grave danger. Study the following 4 figures, which explain the process leading up to drug addiction in 4 consecutive stages. Each shows the change in the state of mind in response to the administration of approximately the same amount of drug as a function of time. The levels higher than the normal state of mind represents the euphoric state, while the levels lower than the normal state of mind demonstrate the depressed state. The arrows indicate the time of drug administration. So you can see the graphs in here. Don't forget that the lines which are higher than the normal state of mind are the euphoric states, and the lines which are lower than the normal state of mind are the depressed states. What can you read of figure 1 and 2? So you compare the first and the second graph. Okay, following their first drug use, drug users return to the drug because of the negative effect it had on them. That's not true. We can't really see anything like that. Right at the end of the second drug use, no bad mood is experienced. That's not true. We can see that bad mood is experienced at the end. The euphoric effects of the second drug use fall behind those of the first drug use. Now, this is something which is true. You can see that the curve is much larger in here than here. The drug use leads to bad mood in both cases. That's not true. In the first one, we can't see anything like that. The second drug use evokes a more powerful positive effect. That's not true at all. So basically, the C is going to be the good answer. What can you read of figures 3 and 4? In figure 4, you can see that the repeated drug use does not lead to withdrawal symptoms. That's definitely not true. We can see that it is basically entirely being located under the normal state of mind. Addiction is caused by the fact that after repeated drug use, even the normal state of mind can only be achieved by the use of drug. That is something we can see on the graphs. Figure 4 shows that the altered state of mind can still be achieved at this stage. No, it is not showing anything like that. According to figure 3, the user is in normal state of mind before the drug use. No, it is in a depressed state, so that's not true. And withdrawal symptoms can also appear in figure 3. That is not correct either, so B will be the answer. Exercise 6. The cellular elements of blood. Write a number of the statements in the appropriate places of the Venn diagram. So you can see here the cellular elements, and the following statements should be distributed among them. So there are 4 or 5 millions of them in 1 cubic millimeter. That is true for red blood cells. They have no cell nucleus in their fully developed form. It's true for the red blood cells for sure, but we also know that blood platelets are fragments, so they don't have a nucleus either. So number 2 will come to this intersection. They are formed in the red bone marrow. All of them are formed in the red bone marrow. The A and B blood group antigens are found on their surface. That is definitely red blood cells. If their number is lower than in a healthy person, it can cause hemophilia. And as we know, blood platelets have an important role in blood clotting, so number 5 will come here. Certain types of them are able to move by amoeboid movement. That's very easy. Those are going to be the white blood cells. And then they are not found in the urine of a healthy person in significant amounts. None of them should be present in the urine in significant amounts. The cellular elements discussed in the task make up only one part of the blood. Calculate the approximate total volume of the cellular elements of a healthy adult. Make sure that you include the appropriate unit as well. So when you answer this question, you have to remember that there are, on average, 5 liters of blood in a human body, or 5 cubic decimeters. And the plasma is about 50% of the total blood volume. It means that the other half will be the cellular elements. So your answer can be 2.5 cubic decimeters. During a lab test, the white blood cell count of a blood sample was found to be higher than the normal. The white blood cells were active and viable. Write down what this symptom can indicate. This condition may indicate an inflammation or an infection in the body, as the exercise is asking one symptom. Basically, I just put here infection. During blood analysis of a patient, iron deficiency anemia was diagnosed. The production of which cellular element is becoming difficult due to iron deficiency? As we know, iron is an important building block of hemoglobin, and hemoglobin molecules can be found in the red blood cells. So the answer here is very straightforward, red blood cells. During blood transfusion, it is very important to ensure that the injured person receives blood belonging to the same blood group as his own. In case of emergency, however, he can receive blood from a donor with different blood group type. Who can he receive blood from? Write your answer on the dotted line. An injured with blood group A can receive blood from someone with blood group O, as we know that group O can donate blood to anybody, so he or she can be called a universal donor. An injured with blood group Rh positive can receive blood from someone with blood group. Now let's see what to write in here. So people with Rh positive blood type can receive both from Rh positive and Rh negative blood, but we have to indicate the blood type that is not identical to his own type. So here we need to write Rh negative. Okay. Exercise 7, Beetle's refuge. There are numerous living organisms involved in the decomposition of the dead vascular tissues of trees. A lot of these species are endangered because their habitat is receding. In their decaying trunks, the many hundred-year-old common oak trees, rare witnesses to times long gone, provide home for diverse communities. These trees are mainly found in pastures maintained with grazing and reaping. Below the cork in the tunnel labyrinth of 2 cm depth develop the larvae of the great Capricorn beetle before they pupate. In the tunnels, filled with droppings of the Capricorn larvae, develop the mealworm beetles. The larvae of the carpet beetles feed on the molted skin and corpses of other insects. The fruiting body of the mushroom lung oyster growing on the cork of the common oak is consumed by the darkling beetles, which in turn is preyed upon by the larvae of the rustic click beetles. On the sunny slopes of the Hungarian mountains, we can still find many hundred-year-old orchards. The rare and old tree species provide excellent refuge for many beetles. These trees also need protection because they cannot compete for light with the trees of the surrounding forest, or they are being replaced with planted forest. The jewel beetles, generally found in the Mediterranean areas, live on the pear trees in an old orchard on the edge of the forest on the slopes of Nasai Mountain in Hungary. The species has never been seen on wide pear trees. The adult form of the great Capricorn beetle is seen in the picture. Name one feature visible in the picture which justifies the fact that this species was classified as an insect. So if you observe the picture, you can see many characteristical features. For example, the body parts, the head, thorax, and the abdomen, or the 3 pairs of legs. But as the exercise is asking us to highlight one feature, I just write here head, thorax, abdomen. Name the type of post-embryonic development typical of the great Capricorn beetle. So basically in the text it is being mentioned that it has a pupa state, and we know that in complete metamorphosis it is actually a phase of the process. So the post-embryonic development is called complete metamorphosis. There are plenty of interactions among the populations living within the body of these old trees. Name the type of population interaction which exists between the following living things based on the text. Lung oyster mushroom and darkling beetle. So let's find it in the text. So you can see it here. Fruiting body of the mushroom lung oyster growing on the cork of the common oak is consumed by the darkling beetles. So this is a nutritional relationship, more specifically it's a predation. Darkling beetle larvae of the rustic click beetle, which in turn is preyed upon by the larvae of the rustic leaf beetles. So this is also predation. And larvae of the great Capricorn beetle and mealworm beetle. Where is it in the text? It's here. Tunnels for the dropping of Capricorn larvae develop the mealworm beetles. So this kind of interaction is beneficial for one partner and it is not affecting the other, which is called commensalism. Okay. Which substance does the larvae of the carpet beetles feed on? Starch, cellulose, chitin, keratin, or calcium carbonate? As we can read it in the text, the larvae of the carpet beetles feed on the molted skin and corpses of other insects. Now, this statement helps us to answer the question. We know that the exoskeleton of insects is made up of chitin, which is a nitrogen-containing polysaccharide. So obviously, this is going to be the good answer for question 6. The Capricorn beetle larvae are involved in the breakdown process of decaying vascular tissues of trees. Describe the function of the xylem and phloem vessels in living trees. So xylem is responsible for water and mineral transportation, typically from the roots towards the upper parts of the plant. And the phloem is transporting organic substances from the actively photosynthesizing parts, from the foliage, for example, of the tree towards the other parts. But to tell the truth, the phloem is capable of transporting both up and down, and the transport is basically called translocation. But here we need to write down the function. So xylem is responsible for water and mineral transportation. And phloem is responsible for organic substances, so the transport of organic substances. Exercise 8, Air-Rich blood group system. Following its discovery 60 years ago, the Air-Rich blood group system has become the second most important blood group type concerning its medical significance after the ABO blood group system. Which component of the blood carries the Air-Rich blood group antigen? We know that antigens are being present on the surface of the red blood cells, so we write here the surface of the red blood cells. The Air-Rich blood group system is determined by two alleles of a single gene. The dominant allele is marked capital D, while the recessive one with lowercase d. The family tree below shows the inheritance of the Air-Rich blood group system in a family. So we are looking at a pedigree chart here that basically helps us to analyze the patterns of inheritance of Air-Rich blood group types within this particular family. And what you need to know is that the circles are always indicating the females and the squares are going to be the males. And in this case, the dark signs are going to be the Rh positive individuals, while the empty ones will be the Rh negative. All right, which part of the family tree provides proof for the fact that the allele of the Rh positive blood group is dominant over the allele of Rh negative blood group? In order to identify the individuals, use their numbers next to their symbols and support your answer by including the genotype of the concerned family members. So in order to answer this question, you have to look at parents 3 and 4 and their offsprings in the pedigree chart. If the parents, so the Rh positive parents, would be recessive, it would mean that all of their offsprings would be Rh positive as well. And that's not the case, as we see that we have two Rh negative children. So this condition is not working. Therefore, we can state that these parents are going to be heterozygous for the trait. And this is the only possibility how actually these offsprings can be present in this family tree. All right, so let's write down what we discussed. If Rh positive allele were the recessive, no Rh negative children, so no Rh negative children would be present within the offsprings of parents 3 and 4. Therefore, we can conclude that Rh negative must be the recessive and Rh positive is the dominant form. Okay. In the family tree, try and find proof for the fact that the gene of Rh blood group system is not linked to the X chromosome. In order to identify the individuals, use their numbers next to their symbols and support your answer by including the genotype of the family members considered. So let's look at the Punnett square again. If the trait was X-linked, then this male would be the following, and the Rh positive mom would have either this genotype or this one. And if you know how to calculate the offspring ratios, basically you can conclude that it's impossible to have an Rh negative female offspring here if you have these kind of parents. Now let's support the statement with the Punnett square, okay, which will indicate immediately that it is impossible to have an Rh negative daughter. Okay, and this is actually D in here. So you can see that out of the girls, all of them will be Rh positive. So this is positive, this is positive. From the boys, one of them will be positive and the other one is negative. So if the trait was X-linked, the Rh positive father couldn't have an Rh negative child. So let's write it down. So if the trait was X-linked, the Rh positive father couldn't have an Rh negative child. Daughter, basically. Rh negative. That's important. Daughter. Okay. Under which circumstances would the Rh incompatibility present a danger? Write the letter of the correct answers in the boxes. In order to answer this question, you have to remember what you have learned about Rh incompatibility. So that's a condition when the mom is Rh negative and the baby is Rh positive. Typically, it's not dangerous for the first pregnancy or for the first baby, but during delivery, the Rh negative and the Rh positive blood get mixed, and that will initiate the mother's body to make antibodies against the unknown substance, against the Rh positive cells. And during the second pregnancy, it can cause severe damage. Now, luckily, doctors can use special treatment in order to basically avoid this kind of incompatibility. So let's look at the answers. The fetus is of Rh positive blood group, while the mother is of Rh negative blood group in the second pregnancy. The fetus is of Rh negative, while the mother is of Rh positive. During the first pregnancy, if the mother and fetus do not have the same Rh blood group, during the second pregnancy if both the first and second fetus are of Rh positive blood group, or during the second pregnancy if both children are of Rh negative blood group. So basically, A is definitely a good answer in here, and B is not. During the first, C is not, as we discussed, the first pregnancy is typically not affected. During the second pregnancy, and this is true during the second pregnancy, and that's not true. Sorry. So the good answers will be A and D. Okay. What is the immunological explanation of the Rh incompatibility? During pregnancy, the blood cells of the mother can pass through the placenta into the fetal blood. That's definitely no. During pregnancy, the blood cells of the fetus can pass through the placenta into the maternal blood. No. During pregnancy, the Rh antibodies of the fetus can pass through the placenta. Basically, the mother's antibodies. During pregnancy, the Rh antibodies of the mother can pass through the placenta into the blood of the fetus. This is true. During delivery, the blood of the fetus coagulates if they do not have the same Rh blood group type. That is not true. So the good answer will be D. And let's start to do the last exercise, exercise 9, with and without light. The students carried out an experiment with pondweeds and a few freshwater snails in glass containers. They use a dye which is blue in water with low concentrations of carbon dioxide and turns green as the carbon dioxide concentration becomes high. A few of their notes are represented below. The pondweeds in the glass cylinder was placed in the sunlight for an hour. The water, which was originally green, turned blue in the container. Then the glass cylinder with the pondweeds in it was put in a dark box which caused the water to turn green again. One freshwater snail was put into the covered glass cylinder in addition to the pondweed, and the whole setup was put in the sunlight for an hour. The green-colored water in the cylinder turned blue. The experimental setup was covered with a black box which caused the water to turn green again in an hour. And the third observation: 4 freshwater snails were put into the covered glass cylinder in addition to the pondweed, and they were all put in the sunlight. The originally blue-colored water in the cylinder turned green in a couple of minutes. When planning the experiment, the students wanted to investigate several relationships. Which two of the following statements support the first experiment? So this part of the text should be analyzed. So the original green color turned blue, which simply means, if we keep in mind that basically the blue color indicates a low carbon dioxide concentration and the green is an indicator of a high carbon dioxide concentration. So the carbon dioxide concentration decreased in this experiment. Now, let's quickly look at the statements. Plants produce oxygen in their photosynthesis. Plants produce carbon dioxide through their cellular respiration. That's true. Plants do not produce oxygen without animals. That is again not correct. Plants die in the dark without oxygen. That's then plants consume more carbon dioxide in their photosynthesis in the sunlight than they produce in their cellular respiration. This is actually explained, so B and E will come to the boxes. What was the function of the dye dissolved in water in the experiment? As an enzyme, it increased the rate of cellular respiration of plants. As a catalyst, it slowed down the rate of cellular respiration of snails. No, it absorbed the poisonous gases of its surface. As an indicator, it showed the concentration of the dissolved gas in the glass cylinder. That is true. It made it easier to observe the oxygen bubbles. So D will be the good answer. Why were the glass cylinders covered during the experiment? So that no dirt could get into the water of the cylinder from outside. Because they wanted to study the metabolic processes of living things involved in the experiment. we will see, so that no nitrogen can dissolve into the water of the glass cylinder from the atmosphere, nothing can be dissolved, because they wanted to study the rate of photosynthesis of the plant in a closed system in order to prevent evaporation. So basically, and the key says the same, that B or D are the answers that could be accepted here. I write D, as it is a little bit more specific for me, but B would also be accepted. Which of their observations proved that plants produce carbon dioxide through their cellular respiration? Give your reason for your answer. So again, we have to go back to this first experimental setup when it is explained that water turned back to green from blue when the glass cylinder was placed in a dark box in the last part. Okay, so this is what we need to write to the dotted lines. So in the first experiment, water turned back to green when the glass cylinder was placed in a dark box. Okay. Which one of the followings is equally characteristic of both the pondweed and the snails? Autotrophic nutrition, that's definitely not. Mitochondria, both have mitochondria. Cell wall, only plant cells have cell wall. Oxygen production, no. And chloroplasts, only plant cells have chloroplasts, so B, very easy one. Which one of the following explanations best account for the changes observed in experiment 3? So in the third experiment, 4 freshwater snails were put into the covered glass with the pondweed, and they were all under the sunlight, and the originally blue colored water, which was low in carbon dioxide, turned green. Okay, so the snails consumed the pondweed leaves. No, there were algae living on the body of the snails, and the algae rapidly multiplied. The pondweeds were not able to set free enough oxygen from the water through their photosynthesis to meet both their own and the snail's oxygen requirement. In their photosynthesis, the pondweeds were able to absorb less carbon dioxide than was released from their own and the snail's cellular respiration. Now this is the case. Okay. And then the pondweeds were shaded by the large number of snails and they did not get enough sunlight. No. How would it be possible to turn the water in the glass cylinder blue in experiment 3? So when we want to turn it back to blue from green, it means that the carbon dioxide concentration should decrease. So let's try to find a statement that will help us to decrease the carbon dioxide concentration in the system. Oxygen should be supplied through a narrow tube. Carbon dioxide should be supplied. No, we want less. By putting the glass cylinder into the dark before taking the snails out of it. A lot more pondweeds should be put into the cylinder and illuminated. Now this is going to help us to get back the blue color because more pondweeds will do more photosynthesis, so the carbon dioxide level will be lower. Therefore, the color will turn from green back to blue. The pondweeds should be taken out of the cylinder while the snails should be left in there. That is not so D. Welcome here. We finished. Okay. All right, so I hope it was helpful. Should you have any questions, feel free to write me an email. And I keep my fingers crossed for you, and I wish you the best of good luck for the final examination.